Hello, everyone. It's my pleasure to introduce our speaker of today's ESDR Kitchen webinar recipe. Um, it's Carmen Gerlach, who is a principal investigator at Karolinska Institute and a professor that she was joining recently at the University of Regensburg at the Leibniz Institute for Immunotherapy in Germany, with this co-affiliation in Sweden and Germany now. So Carmen did her doctoral training with Ton Schumacher at the Netherlands Cancer Institute in Amsterdam, and she was developing there single cell fate mapping technology to track the fate of CD8 T cells in vivo. And she then continued in this direction in the lab of Uli von Andrian. She was actually my bench neighbor there, which was very nice time. And she continued in the field of CD8 T cell diversity and characterized a novel subset of memory CD8 T cells based on the receptor expression of CX3-CR1. And this was published then in Immunity. And she then moved on after a short visit at the University of California, Berkeley, in the lab of Nir Joseph. She then became assistant professor end of 2017 at Karolinska Institute. She was then promoted a principal research investigator there, and very recently also joined as a professor at the University of Regensburg and the Leibniz Institute for Immunotherapy in Regensburg. So in her scientific career, Carmen already made seminal discoveries in the field of CD8 T cell responses and the diversity of CD8 T cell responses, and is also stayed in this field with her own research group and published recently with with CD8 T cells in Immunity 2023 in European Journal of Immunology and Trends of Immunology. And also the title of her talk today makes very much sense in this direction, because she will make sense of the enormous diversity within T cell responses. And I think that's also quite interesting for everyone who is interested in research in cutaneous diseases and in the skin in general. As we know, the T cells are very prominent in the skin. Skin is an immunological organ, and it's going to be very interesting to also translate some of those findings maybe to the tissue and to the skin. So without further ado, I'm very much looking forward to your talk. Thanks a lot, Carmen, for accepting the invitation. And maybe just one comment for everyone who wants to ask questions. This is possible with the Q&A button, and please use it. We will then have a lively discussion, hopefully after the talk. Stage is yours.
Cool, thank you, Georg. Yes, I was very happy. Thank you very much for the invitation. It's nice to see you back, also, even though if it's online. So I just want to quickly check, is it the right screen that is sharing so you see the presentation in full?
Yeah, all good.
Cool.
Very good.
Yeah, so as Georg said, I'm both at the Karolinska and at the University of Regensburg and the Leibniz Institute of Immunotherapy in Regensburg, with the Regensburg site still building up. So if you like what we're doing, I'm actually recruiting postdocs and technicians, so let me know. And also, as Georg said, the interest of our lab is the diversity within T cell responses. And now, if most people, when they think about diversity in T cell responses, they think about the diversity of the T cell receptor. But even beyond that, there is actually huge diversity in the responses. And we know this from studies in, from studies in genetically identical T cells, which were shown to mount identical, very diverse responses. And the diversity we're then talking about is in terms of the function of the T cell, to what degree they clonally expand, how long the cells can live, but also the location of the cells. So into which organ can they migrate? And if they are in different tissues, are they confined to the tissue or are they circulating through the tissue? If they're circulating, what's the migratory route they are following? And all of this is also accompanied by differences in the transcriptome, epigenome, metabolome, and many aspects. More will be, I'm sure, discovered to be diverse also. So, and what I think is important to keep in mind is that we have this type of diversity here within each T cell family. So what I mean with that is within the progeny of a single naive T cell. So in this case, they all have the same specificity and the same T cell receptor. The diversity is also there in the sense that if one looks at different T cell families, they mount different responses. So one naive T cell will give different type of progeny than another naive T cell, even if the same naive T cells to start with were genetically identical and have the same specificity and the same T cell receptor. So why I think it's important that we have this diversity, which is naturally there, is that it gives us robustness to the response, and it also allows adjustability if the cells get into a changing context. So for example, here, if one starts off with a diverse population with cells that have different features represented in the different colors here, if this population gets in one context where feature X is required, those cells with the feature X can take care of the response and the host is happy. If the same population ends up in a different type of context where now feature Y would be required, it would be another subset of the cells, these feature Y cells, that could be able to take care of the response. In contrast, if you start off with a homogeneous population that all have feature X, if this population gets into a context where feature X is required, that's of course wonderful. There will be the response will be very much effective. On the other hand, if that same population ends up in another context where another type of feature is required, the population becomes very almost useless. So therefore, I think it's important to keep these aspects of diversity in mind. For example, if one works in an area of immunotherapy, also vaccination, for example, where I think we need to ask ourselves, what is actually the T cell population that we are transferring or generating or expanding in this particular setting? And also, how do we then get the population that we want? And in order to answer these questions, we need on one hand to be able to accurately measure the diversity. And this is on one hand for mechanistic studies, but also in forms of immunomonitoring. And then we need to mechanistically understand how this diversity or these distinct stages are generated. So we then can exploit this knowledge to adjust the population composition as we need it to get the population that we want. So in red here, these are the questions or the areas where my lab is working on. And when we're coming to measurement of the diversity, we typically do that using subset-specific markers. And that comes because we are operating in this conceptual framework at the moment where T cells are considered to exist in T cell subsets, where each subset in some way derives from another subset, and the definition of the subset is related to expression of certain markers. Now, it is important, I think, to realize that this subsetting framework comes from a time when we were able to not measure very many markers per cell. Now that the times have changed, technological advances are there, we're able to measure many markers per cell or many features per cell, which left us now with this ocean of different T cell subsets. And that then raises some fundamental questions, like what actually should we consider a T cell subset? Is there overlap between some of these subsets? And also, how do human and murine subsets relate to each other? Now, if you think these questions are interesting, then I would like to refer you to this discussion article we recently published on what we're calling the T-cell subsetting challenge, but also to make you aware of these guidelines for T-cell nomenclature, which will hopefully soon, I mean, they will soon be published in Nature Reviews Immunology. This is a huge group of immunologists who has worked on these nomenclature guidelines. But now, beyond even this, we as a group started to ask ourselves if we maybe need a new conceptual framework for this T-cell diversification, one that considers gradients of T-cell states instead of discrete T-cell subsets. And if one now would go from subsets to gradients, we started to wonder, would this maybe help us to translate or to interpret findings between mice and humans in a better way? And the reason we were asking this is because in this subsetting framework, it has largely been unclear how human and murine T-cell subsets relate to each other. And the reason for that is that even though we're using the same names for subsets, so we're calling them central memory cells or stem cell memory cells, effector memory cells, both in mice and humans, we're using a completely different set of molecular markers to define these subsets. And then it becomes very unclear if cells in a human that have the same name as the cells in a mouse actually are the same, do they correspond to each other? Or do cells correspond to each other in both species that carry a different name, but in both cases are the most highly differentiated state, for example? So in this kind of nomenclature problem, I think, is really a hurdle for translation or also the interpretation of findings between studies that go across a species. If one now would consider gradients instead of these discrete subset, would this then allow us cross-species interpretation? And again, the reason why we're asking this is because now that we have high dimensional information from T cells, the technological ability to get this information, we've actually seen that T cells exist in a continuum of differentiation stages rather than discrete subset. So this continuum goes from cells with stem-like features to cells that are highly cytotoxic. And where exactly along this continuum the cells are located depends on a fine balance between different sets of transcription factors. And we now asked ourselves, can we find cell surface markers that reflect this T-cell differentiation gradient in both species? And this is a project that was driven by Anthony Zwijnenberg, a former PhD student in the lab, and had huge contributions of another PhD student, Jyoti Pokharel. And just to mention here, like, you could wonder why cell surface markers if we can go high dimensional. The thing is, you can really not, it's not feasible to always do single cell RNA sequencing, definitely and not in some form of immunomonitoring studies. So we need some very simple measure that still, as good as possible, reflects this gradient, and therefore we're looking for cell surface markers. So the way we approached this is that we took blood from healthy individuals and then performed single-cell RNA sequencing together with antibodies for site-seq, integrated these data, and if one then looks at all antigen-experienced CD8 T cells, so the non-naive cells, that this is indeed a gradient of states bridging the stem-like states all the way to here in yellow, the highly differentiated cytotoxic states. This gradient can be visualized with different colors here in diffusion pseudotime. And now, by asking in order to identify cell surface markers that could capture this gradient, we asked which markers most highly correlate with this graded pseudotime measurement of differentiation. So for that, we're going down from the top scoring molecules down to the first ones that are cell surface expressed and that also have commercially available antibodies for both human and mouse T cells. And then the highest scoring molecule is CX3CR1, which is a chemokine receptor. And these plots here just show that CX3CR1 expression level, or the graded expression of this molecule, is really a nice RNA level graded differentiation marker, because you have a beautiful correlation with the graded expression of these transcription factors that are known to drive T cell differentiation. And you have an equally beautiful negative correlation between CXVCO1 expression level and expression of these transcription factors that are known to repress T cell differentiation and keep them in a stem-like state. So at RNA level, CXVCO1 levels correlate with a graded CD8 T cell differentiation state. So what about the protein level? For that, we went to flow cytometry. And also here you see that there is a broad spectrum of different expression levels of CXVCO1. Now you may notice that we're putting gates here. This is really not to classify them in a subset. This is just a way to be able to quantify the relationship between CXVCO1 level and cytotoxicity as a measure of differentiation. And what you then see is that with increasing CXVCO1 levels, there is an increased expression of these cytotoxic molecules. If one combines this and again looks at CXVCO1 on a graded scale, then you see with gradually increasing CXVCO1 levels, the cells become more and more cytotoxic. With lower CXVCO1 levels, they only express granzyme A. With somewhat higher levels, they start to co-express granzyme A and granzyme B, and only those cells with the highest CXVCO1 expression will co-express granzyme A, granzyme B, and perforin, and be the most cytotoxic cells. So also at the protein level, graded CXVCO1 expression identifies distinct cytotoxic states of these human CD8 T cells in this graded fashion. There is, on more levels, there is actually a relationship between the graded CXVCO1 level and the functional state of these cells in relation to recall potential, so how well they mount a secondary response. We see with an increasing CXVCO1 level, we see decreasing recall potential, which yet again is another measure of saying that with increasing CXVCO1 levels, the T cells become gradually more differentiated. What I thought was most interesting, though, is the relationship between the graded CXVCO1 levels and the production of these effector cytokines. So you can notice here that a single production of interferon gamma, but also the combination of interferon gamma and TNF alpha production is highest among cells with somewhat more intermediate CXVCO1 levels, while the cells that become most highly differentiated, and that's interesting, these are the cells that were most cytotoxic, those cells actually start to gradually lose the ability to produce these effector cytokines. And I think this is a really nice demonstration showing how important it is to look at CXVCO or at T cell differentiation in a graded fashion rather in this discrete fashion of subsets. Because if one would have just looked at your positive or negative for CXVCO1, then you would have come to the conclusion that all CXVCO1 positive cells are effector cells because they are making effector cytokines and they are cytotoxic. But by looking at this T cell differentiation on a much more graded scale, you see that it's not the same cells that are good in cytotoxicity as those that are good in producing the cytokines. So the ability to produce these effector cytokines and the granzymes or perforin are not actually correlated. They're not in the same cells. Now, everything I've shown you so far was from T cells in the blood. Now, T cells in the blood can follow different migratory routes, and we now wanted to know whether the CXVCO1 levels also inform in the migratory capacity of human CD8 T cells, and that is because, yet again, migratory capacity is related to the differentiation state of the T cells. So those cells that have the ability to migrate between blood and resting lymph nodes, or also the splenic white pulp. This is a process that requires CCR7 and CD62L expression. And if we're now looking at the ability of the expression of these molecules in relation to CXVCO1, we see that both CD4 and CD8 T cells, with an increase in CXVCO1 level, the cells lose the ability to follow this migratory route because they are losing the expression of these molecules. When it comes to the circulation through peripheral tissues, here this is yet another route. So some cells may circulate into the peripheral tissues, where they then, together with the tissue-resident memory cells, will survey these tissues, but those, the circulatory ones, are able to go in and out. And as Georg hinted to earlier, what I did in my postdoc in Uli van Anderen's lab is that we actually showed that cells in the mouse with somewhat intermediate CXVCL1 expression levels, they were the prominent cells circulating through the peripheral tissues, while the most highly differentiated, the most cytotoxic cells were unable to enter the tissues, but they were confined to the blood and the splenic red pulp. And we now wanted to know whether that's the case also for human T cells, and again, if this is something the CXVCL1 levels can bridge between these two species. Now, this study here, in order to collect the cells that have the ability to circulate through the tissues, we collected the thoracic duct lymph of these mice. This is nothing we were able to do for the humans, but what we could do, and hopefully this excites you, is that we could turn to the skin. And this is something we did in collaboration with the lab of Liv Aidsmo. And because the skin has these different layers, where in the epidermis, they're primarily tissue-resident T cells, while in the dermis, you have a combination of tissue-resident and tissue-circulating T cells. And we could then make use of that in order to look at the CXVCL1 expression. So if one looks at the epidermis, which has primarily tissue-resident cells, you see that pretty much all cells are negative for CXVCL1. If you're looking at the dermis, and we separate the dermis into circulating and the resident fraction, and we do this by these markers for tissue residency, where we define all cells that lack all of these markers to be circulating, and cells that express at least one of these tissue resident markers, we consider them tissue resident. Like in the epidermis, you see that the tissue resident fraction in the dermis pretty much has no CXVCL1 expression. So this is really just a spillover from the negative population. But if you're looking at the circulating fraction in the dermis, then you see a very clear population of CXVCL1 expressing T cells. Interesting, though, is that the expression level of CXVCL1 here on the circulating fraction in the dermis is lower than the expression level in the blood on the circulating cells. And if one combines all of this together, you can also look at this in this way, where our data show that here with increasing CXVCL1 levels in the dermis, the cells are with more and more likely, they have an increased likelihood here of being circulatory if they're in the dermis. And we also have a mechanistic explanation of that, because actually CXVCL1 expression levels correlate really nicely with S1P receptor 5 expression, and not with any of the other S1P receptors. And Laura McKay has a beautiful study in mice showing that S1PR5 is a molecule that prevents the T cells when they enter the tissue from becoming tissue resident. So this correlation here gives a really nice explanation why, with increasing CXVCL1 level, the cells are more likely, increasingly likely to be circulatory rather than resident. Now, to summarize this, we've seen that CXVCL1 graded expression of CXVCL1, both at the gene expression and then the protein level, really nicely reflects the T cell differentiation gradient, and also informs with relatively high resolution on the gradual changes of functionality of the T cells, with increasing CXVCL1 level, gradually increasing cytotoxicity, gradually decreasing ability to produce IL-2, initial ability to produce the effector cytokines, but with the highest differentiation states, losing the ability to produce the cytokines, and also with a gradually decreasing ability to circulate through the skin and probably even other peripheral tissues. Now, as a little side note, we noticed that CD45RA and RO expression, which are often used to classify human T cells, do not actually measure T cell differentiation, because the differentiation axis here from the stem-like cells to the highly differentiated cells, this is an orthogonal axis to the axis that the 45RO and RA are actually making. So meaning there must be different triggers than differentiation that drive the expression of 45RA or RO. But going back to CXVCL1, what I thought was really cool is that this relationship between CXVCL1 expression level and the functionality was the same in CD4 and CD8 cells, both in the human and in the mouse. And we have looked at all CD8s and CD4 cells in these experiments, though. So with all specificities. And now we started to wonder if it's really such a good translational marker. Can CXV01 be used to even predict the behavior of human virus-specific T cells from murine studies by just looking at CXV01 or using that as a translator? And that indeed seems to be the case. We found this in two very different experimental settings. So on one hand, we looked at virus-specific T cell receptor transgenic T cells in a mouse responding to LCMV infection. Then we had a cohort in collaboration with Sarah Gerlach here in Karolinska with tick-borne encephalitis virus-infected individuals. And in both cases, we sampled the virus-specific cells at different times after infection. And in both settings, only looking at the virus-specific cells in a mouse and a human, we see a relationship where increasing CXV01 levels correlate with increased expression of these different cytotoxic markers. The individual molecules are not the same, but overall the pattern is the same. So increasing CXV01 gives increased cytotoxicity. And this is actually even true in the human here for time points of active infection versus after the clearance of the pathogen. And also when it comes to the relationship to CXV01 expression and the effector cytokines, also there we see the same pattern in the virus-specific mouse cells and the virus-specific human cells, where those cells with the highest CXV01 levels have lost the ability to produce these effector cytokines. So again, as a final summary here, we see that this graded expression of CXV01 marks differentiation states related to different functional states of mouse T cells and of human and mouse T cells, and thereby allows this cross-species interpretation. We've shown this for CD4s and CD8. We have the impression this may even hold true for gamma deltas and NK cells. And overall, we've seen that this T cell differentiation gradient provides a high resolution information on the functional and the migratory state of the T cells, that RA and RO expression are regulated by different triggers than differentiation. And together, I think this has implications for immunomonitoring, where it will help to go from subsets to gradients. I think because of the time, I'm just going to skip the last two slides and go to the acknowledgements to acknowledge the current group at Karolinska, former group members, former students, collaborators across different projects. And as I said before, the data that I'm showing are from Karolinska. We now also, I'm building the group in Regensburg. So if you're excited about what we're doing, let us know. I'm recruiting people there. Thank you.
Thank you so much, Carmen. Very exciting data and very nice to see how the projects developed within the similar field, also with technological advancements and how they build up on each other. So yeah, very nice. Thanks a lot. We don't have any questions in the Q&A at the moment, so please.
No, not right now. I would like to start with more than a question. I mean, it's a comment because, first of all, congratulations. Very interesting work. I mean, you presented different data, different setting experiments. So congratulations. But I was wondering, I mean, I'm a dermatologist, and I was wondering while listening to your talk, in which disease we can apply this model, no? And I was thinking, I mean, the cytotoxic CD8 T cells are important in different skin conditions, but I was thinking at the eczema, and especially atopic eczema, because I mean, there are CD8 cytotoxic cells, and I was also thinking at all the immunomediated therapy that now we use in this condition, because now we are starting to use inhibitor of the axis of OX40 ligand, so very high in the inflammatory cascade. So in the moment of the presentation of the antigen, so it can influence, I mean, the differentiation of T cells. So I was wondering, what do you think of this model in this condition?
Yeah, Thank you. I mean, first of all, I hope I'm not saying strange. So, since I am not a dermatologist, right? I'm a very basic T-cell immunologist.
Yeah,
I have the hope
yeah.
that this as a tool, this looking at CXVCR1 will actually be applicable also in diseases, because we see that even after T-cell receptor activation, we see the similar relationship between CXVCR1 level and the functional state of the cells. Now, we have not looked in dermatological diseases, and I think this is something we have looked at, in a systemic infection at this moment. But I think it is super interesting to indeed look at different types of diseases in whatever tissue, actually, to see if this holds true, because systemically it does. If one does, again, LCMV infection has nothing to do with what is relevant in humans, but it's in a way for us a way to trigger T cells through the T cell receptor, which happens obviously in the disease settings also, but just with a different specificity. Then we do still see the same relationship between CXVCR1 level and functional state. So it would be, I agree, it would be super interesting to look at different disease settings in the human if that continues to hold through.
I also have two questions actually. So I was wondering, I mean, you nicely showed this correlation with S1, PR5, and SIG3CR1. But would it still, do you think, be possible that the T cells in the tissue would downregulate those receptors and become then eventually resident? Or is it just something they are either circulating or they are prone to TRMs, and then that those are two completely different, how to say, lineages or spectra of the continuum, or however you want to call them?
Yeah, I think one thing one should take into account is that the ability for cells to enter tissues from the bloodstream depend, of course, on how inflammatory the setting is at the moment in the tissue, because that influences then the vasculature, and that influences which molecules the cells need to have in order to extravasate and get into the tissue. So under inflammatory conditions, I think the tissue is much more open to and able to recruit T cells of higher differentiation stages as well, that may not be able to continue to sit there if the inflammatory conditions are gone and the antigen is gone because of the S1P05 expression, for example. But at stages when vasculature is relatively permeable to high inflammation, you will have I think you'll have the highly differentiated cells in there too. And then one shouldn't forget also that once the cells enter the tissue, they also adapt on other levels than differentiation. And I would call it a different axis of diversification, because T cells adapt to the tissue and they will change their phenotype. Now, the CXVCR1, since it's on the what I call then the differentiation axis, that isn't really changing, because the differentiation state, once the cells enter, that kind of stays the same. But the tissue adaptation state, that happens too. And some of these markers may overlap and others not. And where we're currently going at is we're trying to dissect all these different axes. So which are molecules that are just driven by differentiation, that are initiated through TCR stimulation, and what are markers that are changing due to tissue adaptation or due to chronic stimulation and so forth. And now I don't remember if I answered all aspects of your question.
Yeah, that's very, very interesting. Yeah, and so my second question was, if you think that this is also an evolutionary concept to keep the homeostasis in steady state, to not let the highly cytotoxic T cells remain in the tissue, but have a mechanism to get them out again under homeostasis.
Yeah, exactly. So if I think this is connected to this ability to enter the tissues and leave the tissues with S1PR5, allowing the cells to leave the tissues. But I think they don't even get in the first place under homeostatic conditions, because the cells no longer express the CXCR3s, for example, that are needed to extravasate, that are not required to the same extent if there's inflammation around. And yes, I totally think that that's a way to protect the tissues from cytotoxic cells at moments when you don't want them there.
Very cool. Yeah. So we actually do have a few questions in the Q&A. One is, does the CX3CR1 follow similar pattern in case of chronic infection like HCMV, and how age affects this expression?
Yeah, so maybe I'll take the age first. So if humans age, the overall expression level, the overall differentiation state of T cells is, as a population, is increasing. And therefore, you also see an overall increase in CXVCR1 expressing T cells with age. Now, we have not done like a formal dissection if the exact expression pattern of CXVCR1 correlates with exactly the same function at an early age and as a late age. I can, like, formally, we haven't tested that. But this overall differentiation decrease in naive T cells and an overall increase in highly differentiated cells with age, this also correlates with an overall increase in CXVCR1 expression in later age. Then with chronic stimulation, there I want to say, so what we have done is in healthy individuals. So these are not people that are chronically infected at this particular moment, but many of them will be CMV positive or EBV positive. So it's a combination of all the specificities that are in a healthy individual, which includes cells that have had chronic stimulation and those that had acute stimulation. And in this whole population, this overall pattern is there. So I think it is definitely useful in this combined setting. Now, if one takes a very specific case of like active ongoing chronic infection to a particular specificity, that remains to be tested. We haven't done that. But what may be relevant to say is that in tumor settings, where you also have chronic stimulation through the T cell receptor, but maybe at lower affinities. But in that settings, CXVCR1 expression is also linked to a cytotoxic state of the cell. So after immune checkpoint inhibition, for example, that is when the CXVCR1 positive tumor-specific T cells are increasing due to their differentiation from the more stem-like cells that are tumor-specific.
Very interesting.
Carmen, we have a couple of more questions. There is one from Anna Dinardo. Keratinocytes release S1P in the barrier, especially during barrier perpetuation. Do you think T cells can enter the skin during those situations?
I don't know, but it sounds very logical.
Okay.
Yeah, and so this may be a way how even the highly differentiated cells can get in through the S1P or 5 expression. But I don't know, we haven't tested. I would think it's likely.
Okay, I will read another one, and the last one I will give it to you, Georg. So, what's the function of CX3CR1 in other immune cells? How will it balance its function among different cells?
Now, I'm not entirely sure if I understand the question right. So
I
maybe
would repeat. So the function of CX3CR1 in other immune cells, and how will it balance its function among different cells?
yes. So I mean, I'm sorry, I understood you, but I'm not sure I understand.
Yeah.
The
I would
person
think
is
I
asking, but
just try to
I'll try
read
to
again.
answer, and maybe the person, if I'm not answering the right question, the person can get back and rephrase, and I'll try again. But in terms of function, so it's a chemokine receptor, and there are a bunch of studies that suggest an effect of survival on the cells, that suggest a migratory effect on the cells. I think there are a lot of studies that aren't carried out in a very good way that I would fully trust these conclusions. We have tried ourselves. We have not found an effect on proliferation. We have not found an effect on survival. There is a pretty good study showing that if CXVCL1 ligand, so CXVCL1 binds to receptor, that facilitates migration through epithelial cells instead of between them. Whether that has a functional consequence in the end, I don't know. It's expressed on monocytes, the receptor also a lot there. It can help them, to attach to the endothelial wall. So it is the ligand is expressed on inflamed endothelium, so there is a likelihood it may help transmigrate, but in vitro studies this has not enhanced transmigration, just the route of migration. So it's a little bit of an open question what exactly the function is. In transwell assays, there is a migration towards the ligand if the cells express the receptor. I hope I answered the question.
Yeah, I think very good answer. Otherwise,
We do.
sure they can contact you also via email. And maybe the last question, I think it relates to something that we already discussed a little bit. So it's relating to the ability of T cells to become tissue resident memory T cells once activated in the skin. And the question is, are all the CX3, CR1 positive cells, intermediate high 1, high 2, able to become TRMs?
Yeah, so here I can answer only with data from other people and some. I started to address this, but haven't come to a full conclusion, let's say. So there are studies again by Laura McKay and I think a few others that looked at KLRG1 expression in the mouse, which is also a marker for differentiation state. So they have sorted KLRG1 negative, which are less differentiated, and KLRG1 positive cells, which are more differentiated, transferred them into mice and checked their ability to form tissue resident cells. And it was the less differentiated cells, which would correspond to the CXVCL1 negative cells that were able to form TRM, and the highly differentiated cells, so the KLRG1 positive cells, were much less able to form TRM. This totally fits with the data of the correlation again of the S1PR5, which drives the cells in normal conditions out of the skin or out of the tissues, and the correlation between S1PR5 and CXVCL1. So even though we haven't done the experiment exactly with CXVCL1, I think the data that are there suggest it's primarily the less differentiated T cells that form TRMs, which also fits with the fact that we don't find this the CXVCL1 expression on the TRMs in skin later on. And it also correlates with the observation that the XTRM, so after re-stimulation in the tissue, that those cells that can leave the tissue and get back in, when they leave, they have a central memory phenotype, so a less differentiated state. So I think when a cell in the skin or in the tissue in general looks more cytotoxic, this is actually a tissue adaptation rather than something on the differentiation axis, where it's I think it's primarily the less differentiated cells that are able to form TRM. They remain lowly differentiated on that axis in the skin, but they can adapt to the tissue, which may include expression of cytotoxic molecules. But it's more of a philosophical question. Like I'm answering, I don't have very hard data. I just want to make clear this, but this is how I think what's going on.
Very good, thank you. Yeah.
I think we are finished. There are no more questions, no?
No, I think
So
those were all the
thanks so
questions. Yeah.
much.
Thank you too. Thank you for the invitation.
Thanks a lot, Carmen. Yeah.
No, thanks so much. Congratulations again. So just to finish, I mean, I want just to remind the next episodes of the SDR Kitchen. There will be another one in 1 month, so the 24th of September, but it will be a surprise because there is no speaker scheduled yet. So freshly baked, really fresh. And on the 8th of October, there will be Martin Roken, and then the Sweet and Sour in November. And we want to remind everybody that all the episodes, maybe in the next slides, there will be the announcement. Yes. that all the episodes of the kitchen, even also this one, will be available, and the next will be available, of course, on the webcast. So they will be free, and everybody can download on the website of the SDR. And last but not least, because it's one of the most important things, in a couple of weeks, we will see each other. So we are all invited. We are waiting for you at the next annual meeting of the SDR that will be in Antwerp, in Belgium. So in less than 2 weeks, we will see there. Very brilliant speaker. We can see in the next slide some of the invited speakers that are going to the SDR. So we can wait, and we can wait to see you there. Thanks to everybody.